Welcome to the deep dive. Our mission today is all about convergence. We're sifting through your sources to find out what happens when these revolutionary anti-aging drugs meet bleeding-edge AI and these new high-resolution biology tools.
Exactly. This is really the moment science starts to move from just theory to highly specific, quantifiable intervention.
We've got three main threads weaving together this deep dive for you. First, we have to talk about a pretty familiar drug category, GLP-1 receptor agonists. And it turns out they're having these profound whole-body age-counteracting effects that go way beyond just weight management. Second, we'll dive into how scientists actually measure this stuff, the sophisticated epigenetic clocks.
And that leads right into our third thread, the tools. Because you can't understand this level of complex biology without equally complex software. We're looking at two really cutting-edge systems, GeodaSuite and CellWhisperer.
Right.
They're built to handle the data tsunami that comes from what we call spatial multi-omics. So integrating everything from genetics to metabolomics and mapping it all out.
Cell by cell, inside the actual tissue.
Cell by cell.
That context is just so vital. This isn't just about a new discovery here and there. It's about making the entire human body, readable, addressable at all these different scales.
Right.
Okay, so let's unpack this. Let's start with the drug breakthrough. Why are GLP-1 RAs, drugs like exenatide, suddenly a hot topic in anti-aging?
Well, it's because of their existing systemic reach. I mean, these are molecules we already know regulate metabolism and energy all through the body.
Yeah.
The sources we looked at highlight a study that tested exenatide, that's exendin-4, not for diabetes, but specifically for its whole-body age-counteracting potential.
So they were looking for something that just works everywhere at once.
Exactly. Something with a wide therapeutic window and a mechanism that hits multiple organs simultaneously, which this drug class absolutely does.
Okay, so how do you test something like that? I mean, aging is a slow, complex process.
They used a really rigorous long-term model. They took male mice, C57DL6 mice, and this is the crucial part, they wait until the mice were already 11 months old.
Which is pretty old for a mouse.
That's considered properly aged, yes. And then they treated them for 30 straight weeks. So this was an intervention aimed at reversing existing decline, not just preventing it from the start.
So what were the results? What were the tangible, observable benefits in these older mice?
The functional benefits were pretty clear right away. The treated mice showed measurable improvements in both physical and cognitive performance.
Okay, look why.
Physically, they just got stronger. They had better forelimb grip strength, better coordination and balance on what's called a rotarod test.
And cognitively?
Cognitively, too. In tests like the Barnes maze and the Y maze, they showed improved spatial learning and memory. It suggests the drug is doing a lot more than just managing blood sugar. It's enhancing these core functions tied to youth.
That's a huge result for a whole organism. But the real deep dive happens when we look under the hood, right?
Yeah.
At the molecular level. You mentioned the transcriptomic analysis.
Yes.
So basically reading which genes are being turned on or off, what did that show?
Here's where it gets really interesting. The analysis showed these robust age-counteracting effects across a stunning number of organs.
Not just the usual suspects like the liver.
Not at all. We're talking the heart, skeletal muscle, the colon, even circulating white blood cells. This is systemic rejuvenation.
And the most surprising part, I'm guessing, were the effects in the brain.
Exactly. They saw significant transcriptomic changes, basically signs of age reversal, in key brain areas like the hypothalamus, which controls metabolism, and the frontal cortex, which is essential for, higher-level thinking.
Wow.
And while they did confirm the expected outcome of reduced neonatal fat, it was this widespread reversal of the aging signature across the board that was the major finding.
So multi-organ, multi-system effect.
Yeah.
But there was a detail in the sources that seems incredibly important. This anti-aging effect was largely specific to the aged mice. Why is that such a big deal?
That specificity is everything. I mean, if the drug was just some general stimulant, it would have similar effects in young, healthy mice.
Right, it would just amp everything up.
Exactly. The fact that the molecular signature, the transcriptome, only seems to get corrected back towards a youthful state in the aged mice, it suggests the drug is targeting something that is actually broken down with age.
So it's fixing a problem, not just boosting a system.
Precisely. It implies a better therapeutic index for an aging human population, maybe with fewer off-target effects in healthy cells. It's correcting dysregulation.
That moves us perfectly into our next segment. If we're talking about correcting dysregulation and measuring reversal, we need a metric. Chronological age just doesn't cut it anymore, does it?
Not at all. We need to quantify biological age itself. At its core, aging is this progressive decline in function that's linked to changes in gene expression. But the key is that this is often driven by reversible epigenetic mechanisms.
Things like DNA methylation.
DNA methylation, histone modifications, yeah. If you can reverse those patterns, you can, in theory, reverse the biological age of the cell.
So if epigenetics is the target, how do you put a number on how old a cell or a person really is? That brings us to the famous epigenetic clocks.
Epigenetic clocks are, well, they're brilliant tools. They're based on measuring DNA methylation across specific sites in the genome. These patterns change in a predictable way over time, almost like a molecular odometer.
And what makes them so powerful is they estimate biological age.
Which is often a far, far stronger predictor of your health outcomes than just the number of candles on your birthday cake. If your biological age is much higher than your chronological age, your risk for all sorts of things goes way up.
Mortality, heart disease.
All of it. Even things like grip strength and walking speed.
Now, when these clocks first came out, the first generation, they were revolutionary, but they weren't perfect. What was the critical flaw that the second generation had to fix?
The first-gen clocks, like the Horvath and Hannum clocks, were mostly designed just to track chronological time. They were great at guessing how long you'd been alive, but not so great at predicting how well you were living.
Ah, so they could miss someone who was really unhealthy for their age.
Exactly. They could lag behind the actual physiological decline.
So the second generation had to bring in real-world health data. Tell us about the two big ones, phenoage and grimage. What makes them so much more powerful?
They brought in clinical relevance. So take phenoage. It doesn't just look at methylation sites. It integrates that data with 9 actual clinical parameters from your blood work.
What kind of parameters are we talking about?
Things you'd recognize: albumin, creatinine, glucose, C-reactive protein.
Markers for inflammation, metabolism.
Right. By weaving in these established markers of organ function, phenoage gives you an age estimate that's much more correlated with your actual health and future disease risk. Grimage is similar, but uses different markers, like plasma proteins. The point is, these new clocks predict functional decline, not just time.
And this precision is what lets researchers actually validate new treatments.
Exactly. We're now seeing measurable reductions in biological age after certain interventions, things like partial cellular reprogramming, using the Yamanaka factor. OSK, right. Or even NAD plus precursors like NMN supplementation. These clocks show they can work.
And the future, based on the sources, is integrating everything.
Yes. The next big thing is the multi-omics aging clock. This isn't just methylation anymore. It's proteomics, metabolomics, all integrated into one predictive model. It's the ultimate holistic view of your health.
Which creates an unimaginable amount of data.
A mountain of data.
And that's the perfect transition. So the GLP-1 array research gave us multi-organ data, the clocks give us the metric, but all this complexity presents a huge data problem. Aging doesn't happen uniformly across a tissue, it's localized. You need tools to see that, which brings us to the Giotto suite.
The need for Giotto comes from the fact that biology is spatial. I mean, a gene turns on at a specific point inside a specific cell in a specific region of a tissue. You can't just grind up the tissue and average the data.
You lose all the context.
You lose everything. So Giotto is a suite of packages designed specifically to handle this spatial multi-omics data. It can analyze it, it can visualize it, and it's technology agnostic.
So it can take data from all kinds of different methods. How does it manage to integrate all that?
It creates this dedicated data framework. Think of it like a highly organized digital map of the tissue. And this map has 3 primary layers of data structures to hold all the raw information.
Okay, let's break down those layers.
The highest resolution is handled by something called Giotto points. Imagine you have the location of every single transcript molecule, every single protein marker, as a tiny dot. Giotto points stores the precise coordinates of every single one of those dots. Subcellular resolution.
Okay, so that's the raw data, the individual points. What about the context? What about the structures?
That's where Giotto polygon comes in. This lets a researcher basically draw boundaries or outlines on the map. These could be the outline of a single cell or even a larger area like a region of inflammation. It gives definition to all those points.
And the third structure provides the visual backdrop for the whole map.
That's Giotto large image. Almost every spatial study starts with an actual image, right? Like a histology stain, an H&E stain, or a fluorescence image. This structure holds that high-res tissue image so you can anchor all the molecular data onto what you can actually see.
What's really game-changing, though, is not just holding the data, but integrating it. You can stack these layers.
Precisely. JADO is uniquely built to integrate data from multiple adjacent tissue sections. So you can take a thin slice of tissue, stack it with the next slice and the next, and reconstruct a full 3D model of the organ's molecular architecture.
Wow.
They showed this by integrating multiple technologies, Visium, Xenium, H&E stains, all on one complex breast cancer data set. It lets you analyze things from the whole tissue domain right down to a single nucleus.
Okay, that covers the spatial complexity. Now let's talk about the sheer volume. The other huge data bottleneck is single-cell RNA sequencing. We're talking 20,000 genes across millions of cells. That's where Cell Whisperer comes in, turning data analysis into a conversation.
Cell Whisperer is really the next logical step. It's a multi-modal AI that lets you have an interactive, chat-based exploration of these enormous single-cell data sets.
You mean using natural language?
Using natural language. You can ask it questions like, show me all the T-cell subtypes in this lung biopsy, and it just gives you the answer with visualizations.
How on earth does it do that? How does it bridge human language and millions of rows of genomic data?
It's built on two core AI models. First, there's a multimodal embedding model. This was trained on just an incredible amount of data. Over 1 million unique cell transcriptomes, each paired with its text annotation from public databases.
So it learns that a specific RNA profile means the same thing as the words astrocyte, frontal cortex.
Exactly. Creates a common language space. Then second, you have the chat model, which is adapted from an open weights LLM. This was fine-tuned on over 100,000 AI-generated conversations about transcriptomes. So it learns the logic of biological questions. It's like a translator that's fluent in both human speak and gene speak.
The validation is pretty impressive. How accurately can it actually tell cell types apart?
Extremely accurately. On the big Tabula sapiens dataset, which covers 24 organs, Cell Whisperer could distinguish 20 common cell types with an AUC value of 0.94.
That's amazing.
It is. Its mistakes were usually only between very, very closely related cell types, which proves it's a fantastic tool for that first pass, for exploration.
And it can be used to analyze really complex processes, right? Like development or disease.
Yeah, look at the chronic inflammation example from the sources. Researchers used Cell Whisperer on a colon data set and just asked for stem cells.
Yeah.
And it correctly identified the LGR5 expressing stem cells, which are a really critical cell type in the gut. But here's the insight. The tool showed that the scores for these stem cells were much, much higher in the non-inflamed samples compared to the inflamed ones.
Which confirms what the original human study found, that chronic gut inflammation hurts these critical stem cells.
Precisely. It validated a complex research finding in seconds from a simple text query. This just fundamentally changes the speed of discovery.
That efficiency is enormous, but it relies on LLMs. So we have to mention the usual caveat.
A crucial reminder. Like any LLM, Cell Whisperer can hallucinate. It might invent overly specific details, like saying T cells from an 85-year-old male when the data just said adult male.
So it's an exploratory tool?
It's a fantastic assistant for generating hypotheses, for finding promising leads, but you always have to go back and confirm the findings against the raw data. It's an assistant, not an oracle.
We've covered a huge amount of ground today, really moving through a true scientific convergence. We started with the GLP-1 RAs, seeing this whole body age counteraction.
And noting that the reversal was specific to aged systems, which is so key.
Then we established the gold standard for how we measure this, the epigenetic clocks, moving from just tracking time to models like PhenoAge and Grimmage that predict real functional decline. And finally, we detailed the software platforms that make sense of it all. JADO Suite for seeing the 3D molecular architecture of aging tissue.
And Cell Whisperer, which uses AI to let us just talk to these vast single-cell databases.
So what does this all mean? Where are we headed?
I think we're no longer talking about, blanket anti-aging cures. We're talking about precision. This convergence of multi-omics and AI is paving the way for highly precise, customized rejuvenation strategies. It fundamentally changes how we model, measure, and then target aging itself.
But that path isn't without risk. The more powerful the intervention, the higher the stakes.
Absolutely. I mean, we have lower risk things like NAD plus precursors, but the more transformative techniques carry real risk. Partial cellular reprogramming has to be so carefully controlled to avoid teratomas or cancer.
And the same goes for things like CRISPR-based editing.
Especially for systemic epigenetic editing. The risk of accidentally turning on a dormant oncogene is very real if the targeting is even slightly off.
So the sources show us the measurement tools are here, the software is ready, we can calculate customized multi-omics clocks, we can analyze tissue in 3D, we can pinpoint where and how an individual is aging. The next frontier seems to be delivery and control. So if we can use these precise measurements to guide the delivery of reversible interventions, like refined HDIC inhibitors or these CRISPR-based editors, how close are we to managing aging not as this inevitable decline, but as a treatable organ-specific condition, something you can manage over a lifetime? What does the application of this technology really mean for individual health optimization when the software can actually guide the therapy?
That is the question.
That's the question we'll leave you with today.